This is the 8th class for your chapter Cell. In the last class, we discussed cytoplasm as well as rough and smooth endoplasmic reticulum, okay, which is the first part or the first organelle and the most important organelle of the endomembrane system. In today's class, we will be discussing Golgi complex, which is the second important organelle or the second in line after endoplasmic reticulum. Let us begin. The term Golgi or Golgi, however you want to pronounce it, it actually came from the name of an Italian scientist called Camillo Golgi. Okay, this guy actually used some dyes and silver stain to find Golgi complex, and it was visible under the normal light microscope. Okay, so this scientist was the one who described its structure. Okay, he was not the first one to find Golgi complex, but he was the first one to describe it. So after him, in his honor, the complex or the organelle is named Golgi complex. Okay. He recognized the apparatus. It is also called Golgi apparatus. It has multiple names. Okay, but in the end, Golgi body, all of it means the same organelle. Fine. So he recognized the apparatus as a reticular structure near the nucleus. Again, the term reticular means a network. Okay, it is a structure which has many networks and is found near the nucleus. It is made up of cisternae. Again, this term is common. Okay, this term we have already studied in endoplasmic reticulum as well. But here also, when you see this, when you see the structure of Golgi complex, you find there are similar kind of cisternae found here as well. Okay, so you can say these are also called cisternae. Okay, these single membraned what you can say tubes or canals are actually called cisternae. Fine. So Golgi complex is also made up of cisternae plus Golgi vacuoles. Now Golgi vacuoles are whatever you see here. Okay, these cisternae are different from endoplasmic reticulum in the sense that their ends are swollen. Okay, these ends you can see are actually bulging. Okay, so these bulges are called Golgi vacuoles. And then you have these small spherical things which come out of this bulging cisternae. Okay, so these are called vesicles. Okay, so these spheres of single membrane, okay, they are called vesicles, same as what we used to call them in endoplasmic reticulum. Okay, so Golgi complex is made up of three parts. It has cisternae, it has Golgi vacuoles, and it has vesicles. In some textbooks, they also mention tubules. Okay, just like endoplasmic reticulum, they mention presence of tubules as well. Fine. It is present in all eukaryotic cells except the sieve tube cells of plants. Okay, remember in phloem you have sieve tube cell and the companion cell. Sieve tube cell is the one where no longer you find any organelles to be present. Okay, so such sieve tube cells, they lack Golgi complex. Then sperms of bryophyte and pteridophyte. Okay, I'll give you examples of bryophytes so you will be able to recall. Moss, club moss, then liverwort, hornwort, all of these are examples of bryophytes. Okay, so they are spores, male spores, which you can call sperms. So these sperms will not have Golgi complex. Then pteridophytes. Pteridophytes are the first vascular plants. Okay, the first land plants you see. So pteridophyte includes all of the different ferns. Then they also include some other land plants. Okay, I'm not able to recall. Equistem, horsetail. So all of these are examples of pteridophytes, right? So in bryophyte and pteridophyte sperm, you do not see Golgi complex. Also in RBC, that is red blood corpuscles of mammals, you do not see Golgi complex. So these are the exceptions where Golgi complex is not found, even though the cells are eukaryotic. Now sometimes the plant Golgi complex is also called dictyosomes. Okay, this again differs from textbook to textbook, but for now this term should be known to you. Okay, what are dictyosomes? They are nothing but they are stacks of cisternae. Okay, so one cisternae is this one, then you have another cisternae. When you form a stack, okay, how you pile up your clothes, when you pile up such cisternae, you can say you have formed one dictyosome. Okay, so dictyosome you can say is without the tubules and the vesicles, only the cisternae, the stacking of cisternae can be called dictyosomes. So dictyosomes are stacks of cisternae that together make the Golgi complex. They are normally stacked very tightly in the animal cells, so one above the other. You can visualize, or you can very well observe these Golgi complex, okay, and they are very tightly organized. So stacked tightly in animal cells, while they are dispersed in plant cells. Dispersed meaning they are not as tightly packed. You will find one cisternae here, then some other cisternae will be a little farther away. They won't be one above the other, okay? They will be a little far from each other. They will be diffused, dispersed. All of these mean the same, okay? They are a little farther away from each other, okay? Far away from each other, making them difficult to identify as Golgi. Okay, if you observe in some plant cells, you will be not able to say exactly that these are Golgi complexes, okay, or these are cisternae of Golgi. The reason being they are dispersed, okay, their dictyosomes are dispersed. Now, talking about number of Golgi complex, or the number of dictyosomes in Golgi complex, it is highly variable. Okay, it again depends on the type of cell and the cell activity. How we talked about rough endoplasmic reticulum, that in secretory cells and metabolically active cells, you find a lot of rough endoplasmic reticulum or even smooth endoplasmic reticulum. Same thing is true for Golgi complex as well. Okay. The number is highly variable. It can be one in some algae, okay, in some algal cells, especially unicellular alga, you can have only one Golgi complex. While in some other, like chara, which is also an algae, you can find up to 25,000 dictyosomes or Golgi complexes, okay, these many. And if you talk about hepatocytes, which are your liver cells, you have 50, okay, in your liver. Now let us come back to the structure. Okay, in this diagram what you are seeing on the left is actually the electron micrograph. Okay, the photograph taken, or the observation, what you can see under an electron microscope, that is shown in this portion, and this is the diagrammatic representation of a Golgi complex. Whenever you see a Golgi complex, let me go back to this previous image. If you see a Golgi complex, you will always find it is a little bit curved. Okay, it has a little curvature. So you will find it is C-shaped almost. Okay, C-shaped on one side and then a little bit bent on the other side. So let us talk about that. The Golgi complex is curved with two faces. Okay, two sides. Sides are here called faces. The convex side, convex is the one that is bulging out, okay, this side, which is bulging out. So this convex side of Golgi is called forming or cis phase. Okay, it is also referred to as cis Golgi, or it is called forming phase of the Golgi, and the network of cisternae here is called cis Golgi network. Fine. Convex side is cis phase. It is the cisternae here are larger in size, they are bigger in size, and they are fenestrated. Remember this term, you will hear this term a lot of times. Fenestration means there are a lot of holes. Okay, it is saying nothing else, but it is just meaning that there are a lot of holes. Holes means fenestration. Okay, so it is fenestrated. It is larger in size and it is fenestrated. On the other end, it is called trans phase or maturing phase. Okay, forming phase and maturing phase, cis Golgi and trans Golgi. Okay, so this here is called trans Golgi network. It is comparatively smaller in size. Okay, that you can see, and it is less fenestrated. Clear? So these are the two phases of Golgi, And with this we end the structure component, okay, of Golgi complex. Now let us see. You can see these small vesicles coming out. These are nothing but spherical sac-like structure, okay, with membrane outside and something filled inside. Okay, we'll find out what it is filled up with. So on both ends you find such vesicles. On the cis side, okay, on the forming phase, you find the vesicles which are coming from endoplasmic reticulum. Okay, the signals, the vesicles, the protein is coming from endoplasmic reticulum. So it is shown in blue color. This vesicle will come, it will fuse with the Golgi complex, and whatever content is present inside will be seen now entering into the cisternae of Golgi, okay, on the forming phase. We will see later on how it is transported and it reaches the trans phase or the maturing phase. Okay. So what are vesicles? They are small sacs of about 20 to 80 nanometer diameter. They contain secretory substance, okay, and are hence called secretory vesicles. Fine. Just look at the diagram here. You have a nucleus. Inside the nucleus, you have DNA. That DNA is being transcribed, which means from DNA you are making an RNA, which is called mRNA. This messenger RNA will exit the nucleus and it will reach till the different ribosomes, okay, which are present on the rough endoplasmic reticulum. So this is the rough endoplasmic reticulum, and on it the green colored ones are the ribosomes. Now these ribosomes, they translate the mRNA and produce proteins, which are shown by these small brown colored dots. This protein moves ahead, and it finally gets packaged in this vesicle, which is also a part of endoplasmic reticulum, as we have already studied in the last class. Clear? Now this vesicle, okay, it is called transition vesicle. This will reach the Golgi complex. Inside the Golgi complex, it will fuse, okay, so the proteins which were made in endoplasmic reticulum are now present inside the Golgi lumen. Here, these proteins are selected where they have to be parceled. So in some ways, Golgi complex is like a post office. Okay, there will be many letters, many different proteins, and the postman, he sorts them out. That okay, this should go to, let us say, Kalavad Road. The other one should go to University Road. This one should go to Lakhajiraj Road. So that is where the sorting takes place. Okay, same thing happens in the Golgi complex. Here also, the sorting takes place. Okay, what protein is to be secreted, what protein is to be sent to the other organelles. Okay. So let us say this brown colored or red colored protein is to be secreted. It is pepsinogen, okay, in your stomach. So this Golgi complex here, cisternae, will bulge on the end. It will get swollen on the end. The protein will be transported inside that swollen end and it will bud off, okay. It will become separated, and this separated form will now be called a secretory vesicle. Okay, and this secretory vesicle or secretion vesicle will fuse with the plasma membrane and the protein inside it will get out. And this is how secretions happen. Fine. So cis Golgi receives the transport vesicles from rough endoplasmic reticulum. Cis Golgi, or the forming phase or the largest portion of the Golgi complex, receives these vesicles from rough endoplasmic reticulum, as we saw here. Then their contents are passed through different cisternae. Sometimes these proteins are modified or they are sorted, okay. And finally, from the maturing phase, which is the trans phase, the vesicles, secretory vesicles are generated, okay, they bud off. Then we talked about the Golgian vacuoles, okay, which are nothing but the swollen parts of the cisternae. So these Golgian vacuoles are expanded parts of the cisternae containing amorphous or granular substances. They also contain a lot of protein or some other substance, okay, whatever has to be packaged. So some of these actually function as lysosomes, okay. So whatever separates from such Golgian vacuoles, it becomes lysosomes. Clear? What are lysosomes? We will study in the next class. For now, just know that how in your body you have digestive system which does the function of digestion. Inside a cell, the digestive system is nothing but a vacuole. Okay, the vacuole is the one which does the digestion process. So it has a lot of hydrolytic enzymes. Okay, enzymes which will degrade or it will catabolize the let us say proteins or sugars or lipids. Fine. So such kind of enzymes are present in the lysosomes, and these lysosomes are actually formed from the Golgi complex. Okay, fine. Now we come to the functions of Golgi complex. The first function is secretion. Okay, all glandular cells in your body depend upon Golgi complex for their concentration and packaging. So all the proteins that are synthesized in endoplasmic reticulum are concentrated. Okay, they are concentrated and packaged inside the Golgi complex. Okay, so as you can see here, let us take this as what cell do you want? Let us take this as a pancreatic cell. Okay, so in the pancreatic cell you have this endoplasmic reticulum. Okay, rough endoplasmic reticulum, it is synthesizing some, let us say, chymotrypsinogen and trypsinogen and carboxypeptidase, sorry, procarboxypeptidase and other enzymes. Okay, a lot of enzymes. These enzymes will be transported through transport vesicles. They will enter the Golgi apparatus. Here they get sorted. Okay, so one let us say chymotrypsinogen will be concentrated here, trypsinogen will be concentrated there. So this way sorting will take place. Okay, after the proteins are sorted and concentrated, they will form such secretion vesicles. Okay, and these secretion vesicles will then finally fuse with the plasma membrane, and the chymotrypsinogen, trypsinogen, procarboxypeptidase, all of these enzymes will be released out into your intestine. Okay, into your duodenum. So this is how the process takes place. Okay, they are sent out of the cells through exocytosis. Okay, this we have already discussed when we were discussing the plasma membrane. Okay, plasma membrane transport. So we discussed endocytosis. In endocytosis there was phagocytosis and pinocytosis. This exocytosis is just opposite of the pinocytosis. Okay, here the vesicles will fuse with the plasma membrane and its content will be released outside. Then formation of membranes. Okay, different membranes are produced by Golgi complex. Okay, and of course Golgi complex is getting its membrane from rough endoplasmic and smooth endoplasmic reticulum. So this is why they are all connected and they all comprise a endomembrane system. Okay, so membranes of vesicles produced by Golgi apparatus join in the region of cytokinesis. Okay, so whenever there is cytokinesis, which means cell division. Okay, so during cell division you require membranes, right? You require plasma membrane, you also require nuclear membrane. So these all membranes are given off by the vesicles which are produced by the Golgi apparatus. Okay, such vesicles, let us consider, which are given off by the Golgi apparatus, they will come together and they will when they fuse they will form one membrane. Okay, so this is how plasma membrane and membranes of different organelles, especially nucleus and all, are made. Then another special role and important function of Golgi complex is it glycosylates. Glycosylation meaning to add carbohydrate, okay, to the protein. If you see here, this is the cis portion. Whatever the proteins are coming from the rough endoplasmic reticulum, these proteins are glycosylated. Okay, they are on these proteins you are adding different sugar molecules. Okay, you can have galactose, then mannose, N-acetylglucosamine, fucose, sialic acid. These are nothing but these are just different types of monosaccharides, and you can see how they are getting attached. If you recall, when we were discussing cell membrane, on the cell membrane, remember how some proteins had those branches coming off and we called them oligosaccharides or carbohydrate moiety. So these carbohydrates added to the protein. Okay, this happens inside the Golgi complex. Same thing with lipids. There are some glycolipids, which means the lipids which have some carbohydrates, okay, with them. Such glycolipids are also made in the Golgi complex. Okay, so protein synthesized by rough endoplasmic reticulum and lipids synthesized by smooth endoplasmic reticulum. They both are combined with carbohydrates to form glycoproteins and glycolipids respectively in the Golgi complex. Then some complex carbohydrates. Okay, complex carbohydrates meaning polysaccharides mostly. So such polysaccharides, then heteropolysaccharides, also modified carbohydrates, they are all made in Golgi complex. Okay, this is very, very important. So all the complex carbohydrates except glycogen and starch, except the two common ones, the two common polysaccharides, all the complex polysaccharides and carbohydrates are made inside the Golgi complex. Okay, it includes the pectins. Remember we discussed pectins when we were talking about cell wall. Okay, pectins are synthesized in Golgi complex. Then you have mucopolysaccharides. Mucopolysaccharides are nothing but they are very sticky material. Okay, they are polysaccharides which give a stickiness. So such mucopolysaccharides are secreted by Golgi. Hyaluronic acid is secreted by Golgi. Then chondroitin sulfate is present in your joints, okay, in your cartilages, in your ear, in your nose, wherever there is cartilage. All those places have chondroitin sulfate. Hemicellulose, again we had discussed in cell wall. So all of these are nothing but carbohydrates, okay, complex carbohydrates, and they are all synthesized inside the Golgi complex. Will you remember this much? It's very, very important. Whatever the carbohydrate you are either adding to protein or lipid or you are synthesizing, this will all happen inside the Golgi complex, okay? Complex carbohydrates. Hormones are synthesized in endocrine glands and packaged in Golgi. Fine. Second thing, matrix of connective tissue. You must have heard of extracellular matrix. Just think of areolar tissue if you can remember or if you can refer. Then areolar tissue has matrix. Matrix is nothing but some watery substance, okay, which is also a sol, and that sol having collagen, fibrinogen, all of these different fibers, okay, elastin present between the cells. So this extracellular matrix of connective tissue is formed by Golgi complex. Fatty acids and glycerol which are absorbed by the intestine, okay, think here. Have you heard of this term lacteal? Lacteal are nothing but they are lymph vessels, okay. So these lymphatic vessels in your small intestine, especially your ileum, they absorb fats and lipids, okay, glycerol and lipids. After they get absorbed, they enter the epithelial cell of your intestine. In the epithelial cell of your intestine, the Golgi complex will package these fats as chylomicrons. Okay, it is very important for you to remember chylomicrons are produced by your Golgi complexes. Then sperm acrosome. Here what you see is a structure of sperm, and the sperm has this top layer, okay, which is called acrosome. Acrosome is nothing but it is a membranous structure which has enzymes, okay, and these enzymes will help it what you can say degrade, okay, or reach till the ovum. Fine. So this sperm acrosome is made up of Golgi apparatus. Then we will study in the next chapter when we discuss cell division. There is something called phragmoplast. This phragmoplast is also nothing but part of Golgi complex, vesicles actually, which are precursor to the cell plate. Okay, when two plant cells divide, there is a cell plate which is formed in the between. Okay, and this cell plate or the middle lamella, okay, calcium pactate, magnesium pactate, is actually made from phragmoplast, which are nothing but parts of Golgi complex. Finally, the root cap cells. Okay, when you study the regions of the root, you will study them in two different chapters. Okay, so when you study the root cap, this root cap cells have a lot of Golgi complex, and these Golgi complex, they secrete mucilage. Mucilage is mucopolysaccharides, and these mucopolysaccharides are actually very sticky and slimy. Okay, so it will lubricate the root tip, and it will allow that root tip to grow deep inside the earth. Okay, deep inside the soil. Fine. So here is where it is important. And the last function in the last part of today's lecture is formation of lysosomes. Okay, we already discussed this in brief, but so there are different digestive enzymes which are present inside the lysosome. Okay, and this lysosome is actually made and packaged and separated from Golgi apparatus. Okay, so some of the vesicles or vacuoles of Golgi apparatus, they store digestive enzymes obtained from endoplasmic reticulum, and such vesicles or vacuoles, they become lysosomes. Okay, what is shown here is the digestion process which I was talking about, that this is a plasma membrane. Let us say the cell has just done some phagocytosis. So phagocytosis, it will take up some food particle inside. Okay, this plasma membrane will close and it will form this vesicle which is called food vacuole. Food vacuole will fuse with the lysosome. Okay, and lysosome has all the digestive enzymes, so when it fuses, the enzymes, they get activated, they digest the food particle, and then that digested small molecules, okay, it could be glucose, fructose, glycerol, fatty acids, they all get absorbed inside the cell. Yeah, so these are the important functions of Golgi complex, and here we end today's lecture. Okay, see you next time. We will be talking about lysosomes. Fine.